microenvironment. Oh, sorry. Live epicyclic, root epicyclic, and endosphere. Invasive species, the association between the symbiotic bacterial and the plant function trait in the context of invasive biology remain unexplored. Moreover, the biogeographic studies of the association between native and invasive population are lacking. And in this study, we investigated the relationship between the symbiotic bacteria and the plant function trait in intercontinental native versus the invasive populations. So our scientific questions were, the first is, are the epiphytic and endophytic bacterial communities differ between the native and invasive population? And two is, what is the relationship between the epiphytic and endophytic bacterial and plant functional trait? Is this relationship between the native and the invasive if have some difference? So we hypotheses were, the first is the epiphytic and endophytic bacterial communities differ between the native and the invasive knotweed populations, and the invasive population have a stronger functional traits. What's more, the benefit more from the symbiotic bacteria than the native ones, as evidenced by the stronger correlations between the symbiotic bacterial communities and functional traits. So to solve these questions, we conduct a common garden experiment in Shanghai. The experiment design includes two populations, the native and the invasive. Each of them have 20 replies the individual from different geographic locations. Totally, the 40 ports. The plant materials used was the knotweed with 20 individuals from China. Japan is the native population, and 20 individuals from the U.S., India, from the U.S., Italy, Switzerland, Germany, and Sweden for the native, for the invasive population. And the soil used in the experiment was 1-to-1 mix of sand and local forest soil from Shanghai. Then we analyze the plant symbiotic bacteria, including the leaf epiphyte, leaf endophyte, and the root epiphyte, root endophyte, and the air, the background soil, by using the 60s DNA sequencing. Additionally, we focused on the 20 plant functional traits related to the growth and the location, resource acquisition and use, and defense. And so here are our results. The first part is the bacterial community composition and the diversity. We can see there were significant variations in bacterial community composition among different host compartments. The bacterial community of leaf and the roots exhibit high similarity, and epiphytic leaf and roots bacterial communities were likely to be derived from the air and the back soil. For the bacterial community composition, is except the leaf endophytes, the bacterial community structures were significantly different between the native and the invasive populations. So this result is consistent with our hypothesis 1. And for the diversity, the invasive population had more diverse leaf epiphytic bacteria compared to native ones. So the second part is the plant functional traits. We can see in general, the invasive population had a stronger functional performance than the native ones, and the invasive population had a higher below ground biomass, higher resource acquisition and use efficiency, and a higher defense. So this result is consistent with our hypothesis 2. For the third part, we tested the bacterial plant trait association. We can see for the community structure, the relationship between plant trait and the bacterial community structures in native population was notably weaker than that in invasive is notably weaker than in native ones. The correlation between the plant trait and the symbiotic bacterial community structures were notably stronger than below ground, stronger below ground than above ground. Then we tested the diversity about the bacteria. We can see the relationship between the plant trait and the bacterial diversity in invasive population were notably weaker than that in native ones. What's more, the epiphytes had a stronger diversity plant trait correlations than endophytes. But in our hypothesis 3, we stated that there is a stronger correlation between the symbiotic bacterial communities and the plant function traits in invasive ones, but our result not correlate with the hypothesis 3. So why is the result? So we think there were 3 possibilities. For the first is the bacterial taxa in our experiments had little to do with the plant functional traits. I.e., there were not bacteria that contribute to plant functions. And the second possible is the invasive population enriched a few bacterial taxa which play important functions, thus reducing the correlation between the plant functional traits and bacterial communities. And the third is the invasive population had a really stronger plant capabilities by itself. And for the plant, it's just invasive, less of them to support their microbiome. So we try to test this possibility. For we test the bacterial texture and functions. We can see the dominant bacterial texture. Here is epiphytic leaf is how to say short-demolars and whatever and the dominant epiphytic bacteria such as the short-demolars are more likely to help plant nutrient uptake and adaptation, where the dominant endophytic bacteria, such as the rhodococcus, are more likely to be involved in plant growth and pathogen resistance. Then we find the significant differences about the bacteria between the native and invasive populations. We can see among the epiphytal bacterial texture with stronger difference between the invasive and native populations. The relative abundance of all texture were higher in invasive, and this suggests that the invasive population are more likely to recruit functional texture of epiphytic bacteria, where the relative abundance of endophytic leaf endophytic bursters and most endophytic root bacteria texture were higher in native than in invasive knotweed populations. And this suggests that the native populations were more likely to recruit texture of endophytic bacteria. This is this part. Then we continue to test the relationship between the significant different bacteria and the plant functional traits. We can see although the different bacterial texture had a stronger correlation with plant traits in native than in invasive, the relative abundance of most epiphytic leaf and root bacteria were negatively correlated with the traits such like the growth, the resource acquisition and use in native ones. And this result maybe suggests that the native population might support those bacteria at expense of reduced growth. It means maybe the result is limited. If they want to, if the native ones want to recruit more this bacteria, it may be a pure trade-off between the growth and the bacteria. So in summary, we can conclude that the community of epiphytic and endophytic bacteria were significantly different between the native and invasive populations. And the invasive populations exhibited significantly higher functional performance, and the relationship between plant trait and the bacterial diversity and composition were notably weaker in invasive population than in native, and we guess it's the possible is the invasive population might enhance their performance due to enrich recruitment of special beneficial epiphytic bacterial taxa, and invasive population investor lies to epiphytic bacteria which are associated with plant functional traits than the native ones. So this is the common garden things. For this, my question is it okay to tell the story like this? And what are the statistical analysis should I do? Continue. And then is the update of the progress of our manuscript in the GRE, and I'm preparing the revision and response letter with the help of Jihua and Oliver, and we plan to submit the revision in a couple of days. So I continue to prepare it. And the last and most important is thank you to our partner for their help. Thanks.
Fabulous.
Yeah, huge congratulations on your nearly accepted paper.
We just
great.
put all those
Yeah,
materials in this afternoon, and just very preliminary results. And we also try to do some analysis like PCA, but still not very successful. So we will do more analysis further.
Good. I have a few comments on your reports. Firstly, as we realized, the Japanese and the Chinese populations are different in many ways. So Japanese populations seems to me more similar to invasion populations, but it's quite different from Chinese populations. So in your analysis, I don't think you needed to take Chinese and Japanese population as source populations. So I think you should separate Chinese and Japanese populations because they're different. That's the first thing. So if you put them together, and then you will get perhaps a wrong pattern of the results. That's the first one. And the other one, you analyze the difference in microbial community of native and introduced populations. The introduced population, the experiments was done in the common garden in Shanghai. So the introduced population to China seems to me are still new populations. So if there is a weak correlation between microbial community and the plant trees. That's reasonable because there is no core evolutionary history. So your explanation should concern the this difference, and also both epiphytic or endophytic species on both invasive and native populations very much depends on the bacterial species pool in Shanghai, which did not reflect the species pool in introduced ranges. So we should consider this the limitation of this of the garden experiments. If we had parallel experiments both in native and introduced ranges, and then you can directly make comparisons. But in this case, we should consider the limitation of our garden experiments that was just conducted in a single site. Yeah, first I have two comments on the results you had so far. So perhaps we needed to reanalyze the data.
So, Bo,
And
when
yeah.
you're talking about the populations in China, are you thinking about the northern populations in China are different from the southern? Because we've talked about the native moving north or being transplanted, and maybe there's a difference between those.
Yes, we all choose the native ones.
Ah, okay.
Yeah.
We should separate both northern southern populations. Depends on how many populations used in Chinese sites.
Because we used 10 individuals from China, 10 individuals from Japan, and 10 from US and 10 from Europe. So in the beginning, we want to compare the populations from 4 regions. But unfortunately, because the results is quite mixed and we cannot find a very clear pattern. So in this version, Jinwen just want to compare the native ones and the invasive ones. We're struggling with this for a long time, actually.
Did you find that there was a difference between Japan and China already, or just between those two, or did you try that?
Maybe Jiwon can show some figures, some previous figure.
Okay, I will finding this, and I also send the map, the symbol map in our chat. The picture. So, can you see the?
Yes, we can see it.
Okay. This is the last year. We divide the four group. the China, Japanese, US, and Europe. But for us, why we choose the invasive and the native? Because it's just divided by human. For us, the Asia's part is also the native ones, and the US and the Europe population is all of them invasive ones. Why we divide this? Just depend on
no,
the
you just show some results, okay?
Okay. Sorry. This is the last video about we divide to the this is this is the first video we divide into the four the China, Japanese, US, and Europe.
I think Lei has a question or
Yeah,
comment.
Lei, do you have something?
Yeah, can I show you two pictures, Jingwen? In my transcriptome PCA analysis, maybe we have some comments here.
Okay, thank you. I will stop the share.
For if you want to combine the Chinese populations and Japanese populations, here I think we should isolate it, because first for their chloroplast analysis, the haplotype in Japan and Chinese, they are different, but the haplotype in Japanese populations and the Euro population and the North American populations, they are the same haplotype. And second, in my transcriptome analysis, here I drawn two pictures yesterday, and I think I should show you this. Can you see my screen?
Yes.
Yeah.
Yeah. There are two pictures. For the left, here is almost 100 individuals from China. The red is North China individuals, and the green is South. The red is South China individuals, and the green is North China individuals, and E for Europe and G for Japan. This is a transcriptome analysis, and I can't put more management on the plant. So I think the Japanese and the Europe individuals, they are combined
More similar.
Yeah. So but Chinese, they are different from these two.
Yeah.
So I think you should put the Chinese individuals and Japanese individuals apart. And the right pictures is Homer analysis. They are the same individuals, but one for transcriptome, but the right is for the Homer analysis. For the same level, I think they are the Europe individuals and the Japanese individuals, they also combine the tight, but the Chinese individuals, they are in the other side. So from these two pictures, I think Japanese individuals and Chinese individuals are not put the same. Yeah, I think. But in your result, maybe they have they don't have large different. But I think you should
I
apart
get your suggestions. We just we really talk a lot of about these problems. It's really hard to solve this problem because the Japanese population is so small. If I put the Japanese population together with another 3 populations, it looks like a point compared to the latitude. So it's really hard to solve this problem. If I divide into 4 parts, but it also have another problem is the number of Japanese knotweed is so limited.
Our background is so different.
So I really don't know how to solve this problem. And sorry, can I share can I share an article about? So we have read this article. Maybe it looks the same with our problem, but so we just have a try to compare the native and the invasive ones. But it's still a big problem. So I want to ask our partners comment about this problem, how to solve. Because we can see the Japanese population is really limited, just a point compared to the other 3. So
Jingwen,
yeah,
we were asking you to share a PDF of your presentation because your results are rather complicated. So it's not easy for us to fully understand your results.
okay.
You can make your PDF PPT file as a PDF to share in the to share it with us.
Okay, maybe after the meeting, I will prepare the PDF for the videos for us.
Yeah.
Okay. Thank you so much.
Madeline, did you want to say
You just put your PDF in the chatting room.
Yeah, if we could go back to your slides for this PCA of the microbial composition.
You mean this one?
PCA.
The PCA, where's the PCA?
Is
the one where you have the endophytes and the epiphytes and roots and leaves.
Mm-hmm.
So I think it is interesting to know about the Japan and China populations and how they are different. But I also still like having or being able to discuss native versus invasive. And I was thinking that when you show I think it can be a twofold comparison, like native versus introduced, and then really see where the clouds are, like if the introduced are overlapping with the Japanese populations, and also to see how much variation you actually capture with those fewer Japanese populations. So, and to begin, or I should have said this at the beginning, maybe. I mean, it's an amazing data set. I think it's really cool to see these results, and it's a lot to think about. I agree with that, but overall, I liked a lot the logical flow of your presentation. I think it's a good structure.
Thanks, but it's just like the Professor Bohr says, it's need to consider the PCA.
Oh, can you go back on the slides, please? I saw the graph that I was trying to get to. I think it's maybe page 10 or something, or if you go on the previous slides, even more. I think it's 10 or earlier, even more up. Yes, yeah. Slide 9.
8.
8. Yeah.
8.
Yeah.
Yes.
Yeah.
Do you have more comments on this, Madeleine? Or
no, I was just looking.
Can I go?
Because go ahead.
I just have a few. I mean, it was a lot of information. First, I agree with Madeleine. I actually enjoyed your presentation quite a lot. The figures, I think the experiment was cool. And the sample size is the limitation, as you already know, that you have these 4 times 10 individuals, which sort of limits your possibilities for making smaller groups and testing for differences. That, of course, is the limitation. But I agree with Madeleine that it was a nice flow of presentation. The figures are beautiful. And also some of the results are very interesting. There were just too many results. I couldn't process them yet. And I think some of the earlier results like this are very, very clear. And I think there is also a difference between native and invasive populations. The only question is, what does it mean? And also, I agree with Bo, of course. What does it mean if you find this now in Shanghai? Yeah, like, because this is the place where it has been tested. But in general, I found this very, very well structured, and the data are for sure interesting, and they mean something. Like, for instance, if you find a significant difference between native and invasive populations, and the native cloud is consistently different from the other, although we already know that Japanese and Chinese populations are genetically different, then you then you know that so these genetic clusters cannot be important determinants of the bacterial structure or something. So it's good to have this information, of course, when you want to interpret things. I have a small, and I will take a look at your PDF later if I can, and maybe think a little more about if I have some useful further feedback. I just have one small technical question. it was too quick at the beginning. So the experiment was only spontaneous colonization of bacteria, right? So you just planted the plants in the garden, and all the bacteria that occurred was spontaneous colonization, right?
Yes, we plant in the common garden and have it in the August, not the other treat. Yeah, just plant.
Okay, so that means the roots were colonized just from what was in the soil there.
Yeah.
And the leaves were colonized from what was in the air at this location. Yeah.
Yeah.
And
the soil is normal to all of the knotweed population.
Yeah. Good, thank you.
Thank you for your suggestion. I will prepare the PDF ones and send her.
Yeah, I just have one more comment that I think would be interesting. I don't know very much about soil microbes, but you mentioned that there are beneficial symbioses and maybe there are pathogenic symbioses. I don't know if it's possible to think about your taxa in terms of positive effect versus negative effect, right? If you have, because you have so many different taxa there, and you have differences between what's colonizing the native versus what's colonizing the invasive. If there's some function there, I think you're really starting to think about this. What's the importance of different taxa? And I don't know if there's more story there, but that's really fascinating. If you're finding that the native plants are colonizing with taxa that are more beneficial to them versus the invasive, but it seems not, because your invasive plants are growing more. Or is there some kind of general avoidance of allowing these bacteria to colonize? So I think the way you're thinking about it is really interesting, and you just have so much data. Maybe it's difficult to find those important signatures, but that's really
Thank you
interesting
for your suggestion. Yeah. We will try to analyze this. Thank you so much.
And I think with your slide 17, it might be nice to see the endophytes across everything, because then, for example, you see these big chunks of color that are in the same compartment, but they change size across the native leaf and root and invasive leaf and root, right? So just different ways of thinking about how to look at these data to see if there are patterns. That's what I mean, I agree with Oliver. There's so much information here. It's really hard to digest. And I don't know anything about any of these taxa, if some of them are supposed to be good things and others are bad things, and maybe we don't know, but
I have a quick comment. Since we're looking at this slide, maybe it would be nice to group them by organ, or this epi leaf. Have the epi leaf native next to the epi leaf invasive.
Yeah, exactly.
You could easily track the changes.
But I think, this is a tremendous amount of information, and it's a really, I don't think you need to put everything there, because even just the first part of the story is so interesting and new and exciting for the field, that even just the first part where you're showing the basic differences between the native and the invasive is an awesome story. And I understand that now you're excited and you want to track it down a little bit more, but it might be just the easiest way to get a really nice, important paper to have this first, wow, there are these big differences. And we found that they are related to the landscape somehow, or they're related to native versus invasive on a big scale. And then the next paper can be more like, we found this before, now let's drill down into it. So that's one way you might think about how to tell this first story. This is just the first story. You're not going to answer everything, but this first story is really exciting just because it's the first time we know anything about the differences between these groups.
Thank you so much.
Did you have
Yeah,
some?
I agree. I agree with that. You should simplify your story to make it a coherent story, not a scattered story. If you write your dissertation, you can put everything in your dissertation, but for a single paper, you should have a simple story, especially to show you normal results. That's important.
Oliver,
Thank you so much.
I have one final comment. But I don't know, Madeline, maybe you have to help me because this is stuff that John He is doing. Can you do some kind of functional analysis or functional trait analysis of the bacteria? Or is it all just bacteria or is it also fungi? No, it's bacteria, right?
Bacteria.
I mean, because we were just wondering who is bad and who is good. Christina asked that also. And isn't it possible? I mean, aren't there like functional trait databases for bacteria, taxa, where you can say, okay, there, and which ones have been associated to be pathogens or not, or mutualist or not, and then you can maybe simplify this, you know, from taxonomic, many different taxonomic group, and just boil it down to, I don't know, frequency of being associated with being pathogenic, and how does that differ, like the share of potentially pathogenics on the different types or so.
But this is something that Yun He did, but also Max together with Sigi.
Yeah.
So
it can be done. I know several other, we're in several other projects where this can be done. So you turn the taxonomic grouping basically into a functional groups, and then it becomes a bit more ecological also, and not so confusing.
Okay, I will ask her.
You can write us an email, and you can write us an email, and we can try to, if you don't know this already, we can try to connect you with some people who can tell you which software and which package or so to this, how this can work.
Okay, thank you so much for that.
And I wanted to see again maybe this figure where you see these two overlapping clouds, like natives and invasive, and I think it's on slide 9. Can you go to that again? Okay, so here, for example, this would be a way to solve this Chinese, Japanese populations. I think you could just mark them differently, or even try to draw the ellipse. So still have this native versus invasive in blue and red, but maybe change the shapes and even try to draw the ellipse for the Japanese populations and the Chinese populations separately. But yeah, while having these, I think this would be great. And in fact, I find this result very cool and fascinating.
Thanks.
So
I have a question. So you measure the bacteria in the plant, right? And also in the background, in the air, and also in the soil, right?
No, the air is plastic leaves.
Plastic leaf. Okay.
Yeah.
I'm wondering, is it also some difference in the in the rhizomes at the very beginning. So maybe it would be interesting to also measure the bacterial composition in the rhizomes for the experiment. Yeah, so it should be cool to have that result.
Mm-hmm. And before we use that, we have to this.
Yeah, but maybe some endophyte bacteria can survive.
Yeah. It's maybe, but it's really different for the, you can see the endo and the epiphyte. So we can see the endophytic is quite similar. Not only the native, but also the invasive compare the epiphytic bacteria.
Yeah, it's too bad we didn't have the information from the plants when we first collected them. That would be really interesting.
Jingwen, for the finger 5, I have little tips. Maybe if you could put the finger 5.
This one.
Yes.
Okay.
Put the for example, EPI L native and the invasive, these two put together. Maybe we could see it visualized. Yeah.
Okay, nice, good idea. Thank you so much.
For example, 1 and 5 put together and 2, 6. Yeah.
Okay. Thank you so much.
The
I will do that.
picture is beautiful.
Thank you.
Great. So I guess we can continue by email to help you with the interpretations as you go. So we've already run through a full hour, and I know everybody's got a lot to do. I don't know if Pepe was hoping to present anything today or if somebody else had something in mind, but we could probably try to squeeze one more.
Hello? One don't like to present his results.
Hi, great.
Where he obtained in Kaifeng. Yeah, Wan Dong, can you very briefly talk about your results?
Okay. First,
that you obtained in Kaifeng Common Garden experiment.
Okay, thank you, everybody. Thank you very much for giving me this chance to share my experiments, my research carried out at Kaifeng. And to be honest, this is my first time to talk here, but I learned lots of knowledge from everybody here. Thank you very much. Before the report, please allow me to make a brief self-introduction. Okay, my research field is ecological and evolutionary interaction between plants and insects and biotic stresses. And in the past 4 years, I joined Professor Dean group and especially focused on the effects of repeat evolution of herbivore defense on other trees, such as biotic stress tolerance in invasive plants after reassociation with the specialist insects, and by using the worldwide invasive plants common ragweed. Okay, let me start my. Okay, because of the plant limits at Kaifeng, we only have 2 Japanese knotweed populations, Chinese population and Europe population. Each population we have 10 from 10 sites and 40 individuals. This year's Carbon Garden experiments. April, we cultivate the plants, and in May, we recording flowering rate, plant height, stem diameter, leaf toughness, leaf surface area, and sampling leaves for chemical analysis. Sorry. In September and October, we carry out experiments at the plant's mature stage. We record above ground plant height, ramming number, leaf damage by herbivores and pathogens, taking photos of all plants and collecting above ground plants for weighting, leaf weighting, flower weighting, and stem weighting. Then we collecting and weighting the below ground plant tissue. This is the picture of we did this experiment. To be honest, this part I think is a very challenging task because the cultural medium is tightly attached to the root rhizomes, so it's very challenging. And look at the results. This is results about at the growing stage. We found that the Chinese population, their height is higher than the European population. Then the physical defense, we collect the leaves and measure their toughness and thickness. We found that the European population, their leaf toughness and leaf thickness is larger than the Chinese populations. The chemical defense here, we only measure 3 chemical compounds, and others we haven't finished. And look at the leaf tannin. We found no significant difference. The same is the alkaloid, but the leaf lignin, the European population is larger than the Chinese population. Here, sorry, I didn't analyze them. I only show the tradition. Look at the results of the plants at the mature stage. We found that similar with the growing stage, the Chinese population, their height is higher than the European population. Look at the picture when we collected. And this is the more significant difference between the Chinese and the European population. This is the number of rameys. The European rameys number is significantly larger than the Chinese population. Look at the herbivore damage. We found that the European population, their percentage of chewing herbivore damage the leaf area is smaller than the Chinese population. Then the patient infected plant leaf, this is similar with the herbivore damage the leaf. The European population is smaller than the Chinese. Look at the pictures. This is the Chinese population. Is the person that is very seriously. This is the leaf bios and stem bios. We found the leaf buds, they don't have significant difference. Look at the, but the flower and stem buds, the European population is lower than the Chinese population. This is a below ground plant tissue, rhizome biomass. Chinese population is higher than the European population. This is similar with the fine roots. The Chinese population is higher than the European population. Okay, take home message. In comparison with the Chinese population, native population, the Japanese knotweed from European population have relatively high defense against the biotic stress herbivores such as herbivores and pesticides, but having low growing performance, such as their above ground and below ground tissues weight. This seems to contradict the EICA. Second, the enhanced defense in Europe population was due to the increased resource allocation to physical and chemical deficit. Here, the leaf toughness, leaf thickness, and such as lignin, maybe some other chemicals we haven't finished the preparation. But at the cost of the resource allocation to rhizomes, we found the rhizomes, the Chinese rhizome biomass was heavier than the Europe. Third, the invasive mechanism of Japanese knotweed in Europe was primarily caused by the enhanced number of ramis and the enhanced biotic defense. I think because the ramis is very different between the Chinese population and the European population. The next is experiments in 2022, the last year. Here we, according to the Fudan, give us the procedure. We insect sampling insects using a large trap. During and July, we recording some index. That's the most of last like in 2023. Here, a new one, we recording the leaf photosynthetic rate and some other index using the portable photosynthetic equipment. Then at the mature stage, we at October, we're recording plant height and the male-female monocysts plant, taking photos of all plants and collecting above ground plants for weighting. Look at the results of the knotweed at the growing stage. And for first, the plant weight and the diameter. I like the results in at the 2023. We found the height didn't have a very significant difference, but the diameter, the Chinese diameter, was larger than the European. The number of ramis similar to above. The Europe ramis is larger than the Chinese population. This result is similar with the branches. We look at the leaf number. We counted all the leaves in each plant and found the there was no difference, no significant difference between the Chinese and the European population. But the European leaf, their fresh weight and their dry weight is heavier than the Chinese population. This is very significant. We also scan leaves, but my student didn't give me this data. They forget to calculate the areas. Maybe soon we will finish it soon. Look at the herbivore and pathogen infection. This is similar with the experiment results in 2023. The proportion of herbal damaged leaves, the Chinese population is higher than Europe, but the percentage of herbal damaged leaf area is not a very significant difference. But the percentage damaged leaf area, the European population is significantly smaller than the Chinese population. This is the leaf chlorophyll content and the photosynthetic rate. I expected that the Chinese population, their chlorophyll content is higher than the European population. And this is similar with the photosynthetic rate. The Chinese population photosynthetic rate is higher than Europe. Okay, I only prepared the growing stage. We also have some results at the mature stage. Maybe the next time I can show them. Take home message. This is similar with the results at the 2023. Okay, this is my 2 years results. Thank you very much. This is Professor Ding and got me and lots of students who help us to finish these experiments. Thank you.
Thanks, Wan.
Thank you.
And Wendell, thank you. You got lots of results.
Sorry for my terrible English. I'm not sure if you understand me well.
Perfect. No problem.
Okay.
Yeah,
Yes.
perhaps you can put the 2 years results into a single story, maybe, if you didn't make a comparison between years.
Mm-hmm. I think it's really fascinating that, so we have the same story. I don't know if you've seen Pepe and Shengyu's data, but they show the increased ramette production also in the invasive populations. So it's in all of these studies, we have a lot of support for more ramettes in the invasive population. So that's very cool.
In this sense, I'm very interested in the genes that's associated with clonal growth. Yeah, clonal growth.
Yeah.
Yeah.
I don't know anything about that, but we should think about it.
Yeah, ask later. Do that. Yeah.
Yeah.
Nick, can you talk this? Yeah, that's very interesting because
it is. Yeah.
Yeah, because in both kiloweeds and the Subatina, we had similar results. The invasive populations normally have higher clonal growth rate than native populations.
Yeah.
I think this is very important for a species to separate horizontally, especially locally, and also this will make the invasive species more competitive than the natives.
Okay.
And also, Zhiyong, you had similar results in Xishuangbanna?
Yeah, I just finished the common garden harvesting several days before, and I had the data yesterday. I can show you quickly some of my results. It's quite different from the results of from the other gardens. Yeah.
No!
You got different story.
Okay, I will show my screen.
Okay.
Mhm. Okay, so some updates from the XDBG Common Garden. So firstly, an overview of the traits I collected in last year. No, in this year. So for plant growth, I measure the plant height and the remit number monthly. And also I measure the leaf thickness and chlorophyll content in middle of the growing season, and also the phenology. And I also monitor the plant hybrid damage twice during the experiment. And for the harvest, I measured, I harvest the above and below ground, and I also separate the above ground to leaf, stem, and the reproduction parts. And for the below ground, I separate them to rhizome and the fine roots parts. And I also take 2 photos for each individual, one from the side and the other from the top. And this photo shows you what we were doing. So cutting the background biomass, and this shows at the end of the experiment, all pots gone. So we harvest the background biomass during 8th September to 12th, and harvest the below ground biomass during 15th to September to 11 October. And this photo shows you how we did. So we cut the plant down and separate them to different parts, and then weigh the dry biomass. And for the below ground biomass, it was really challenging, but we did that according to Professor Bo's suggestion. Actually, they did some pirate experiment. Several Professor Bo and Wu, and also Ju. So for each plant, we cut them into 2 parts, like the small part and the larger part. And then for the small parts, we wash the rhizome and also the fine roots. And for the larger part, we wash the rhizomes only. And then for each individual, we select a sub sample, like 8 rhizome pieces for new experiments in the coming year. Yeah. And we wait the rest of the rhizome. Oh, no. We put all the rhizomes into a pill bag, and then we oven dry them to constant weight, and then weigh the dry biomass of the rhizome and also the fine roots. So this picture shows what we did. We set up a working flow, like with a group of people. So it was really really funny, actually. And as you can see, this is actually at the very late of the harvesting. Some of the plants already regrow out, and but this is a very small part of the biomass compared to rhizomes. I put them into the rhizome parts. So this photo shows you like the small parts, what we had from the small parts, and this is what we had from the large parts of the substrate. So this is what we were overdry the biomass. So we use several large ovens and quite many biomass, and we weigh the biomass. Yeah. So for the results, and I only show you the very preliminary results. So I just had them 2 hours, actually 1 hour before maybe. So I used linear mixed effect model and put the initial rhizome weight as the covariant, and I used the range as fixed effect and the population as a random effect. And this is the rhizome differences across different ranges. So that's why I said that was very different to the other experiment. So that we found that the Chinese populations, they have significantly lower rhizomes compared to the other 3 regions. And this is the post hoc comparisons, like the Chinese to European, Chinese to Japan, and Chinese population to the US populations. They all differ significantly. But we didn't detect significant difference among the other 3 regions. And for the fine roots, the Chinese populations had significantly higher fine root biomass compared to the other regions. So that's the very general pattern. And I also have a look at the within range variation for the below ground biomass. So the upper plot is the rhizome biomass, and the lower plot is the fine root biomass. I didn't forgot to change the label. So as you can see, for the Chinese populations, I plot them along the latitudes. So they were very huge variations among populations. And for several populations from the northern part, they were quite different compared to the southern populations. Maybe that's also I think consistent with the our other findings. Yeah, this is the fine roots. And for the rhizome biomass variations among the European populations along the latitudes, they were less variable. Yeah, so this is the Japanese populations. Yeah, also less variable. And this is the result from the US. They are also quite huge variations. And yeah, I think that's the very preliminary results. Yeah, I think in the future I will also try to correlate the results with the other data. Yeah, and such as the climate and also the herbivore damage. And actually, the herbivore damage was also quite different to the other 2 gathers. Yeah, yeah, that's it.
Great, that's cool.
That's great. Yeah, great results.
Yeah, and it's cool to see that you're still finding the real similarity between Japan and the invasive populations. That seems a really strong, consistent message we have in our data.
Yeah, and also, yeah, and also for the total biomass, I think the pattern was quite similar to the rhizome biomass, because the rhizome biomass account for huge proportion of the total biomass.
Yeah.
So that's I think in XTBG common garden, I think it's consistent with the ICO hypothesis. Yeah.
Yeah. Okay. Interesting. Yeah. All right. I won't want to cut everybody off, but it is late and I have to run. So we're going to have to pause here and pick this up for next time. I hope that's okay with everybody. People are running off.
Yeah.
I have to get my son to school and get myself to school. But this is really great, and I think we're really amassing a lot of awesome data. And next time I think we will, so maybe I'll just send out an email to the whole group and find out if some people were unable to present today that would like to next time. And we'll also try to get short presentation from Isolde and from Lei on the genomics if we can manage to still fit everything. That's the strategy. That's the plan anyway. I hope we can do that, but we might not have enough time as usual.
Yeah, I think we needed to think about how to put the results from all three gardens together.
Absolutely.
To make a story, to make a single story rather
Mhm.
than three different stories, because we actually share the similar patterns anyway among the gardens, or
Yep.
some of the traits to variables. So Zhiyong, Wandong,
Yes. Yeah.
and Jingwen Pepe. So
Mhm.
we should think about for all the three gardens.
Yep.
Yeah.
Yeah, it's a
Yeah,
nice
can you
story because most people don't have common gardens in so many different places. So it's nice to see that how different they are and how similar
Yeah,
in some cases.
Yes,
Yeah.
yes, it's very important.
or similar or whole difference
Yeah.
in terms of the difference of climate variables at different sites.
Yes.
So obviously, Xishuangbanna is a tropical sites, and
Mhm.
Kaifeng is typical temperate sites. So Shanghai is tropical. So our experiments actually conducted across three climate zones.
Yeah, exactly.
So if the results are different, that's okay. If are similar, that's okay. Also okay.
Yeah.
But we need to analyze why that.
Mhm.
Okay.
Great idea.
Yeah,
Okay.
Jiyoung, can you lead this?
Yes. Yeah.
Awesome.
Great.
Yeah.
Cool.
Yeah.
All right, I gotta go. It was really great to see you guys. And
Okay,
we'll see
so
each other in
see you.
December.
Okay, maybe
Yeah.
okay,
See
see
you.
you maybe next month, just
Okay.
before Christmas.
Yes.
Okay,
Great.
yeah, okay. So
Yeah.
Thanks, everyone.
nice to see everyone. Okay, bye-bye.
Yeah.